So, welcome to this class on Neuroscience of Human Movement, which is on primary motor cortex. This is part 3. So, in today's class we will be reviewing content from previous classes. So, what was discussed in the previous 2 classes at least, and we will discuss the role of premotor and supplementary motor areas, and intracortical microstimulation and recording techniques, how these experimental approaches pioneered insights into the way motor cortex functioned, and how individual pyramidal tract neurons are responsible for movement generation, especially the works of Dr. Ed Evarts, right. So, to begin our review, first we discuss the work of Campbell and Brodmann, right. So, Campbell and Brodmann classified cortical areas, classified the structure based areas, or in other words made cytoarchitectural classification, and they found that the area that is immediately rostral through the central sulcus had distinct structure, in the sense that it lacks. So, this area is now called as primary motor cortex or M1. M1 lacks the 6 layers that is characteristic of the cortex, right. Cerebral cortex generally has 6 layers, that it is also granular, contains the granule cells, but this granular structure was absent in M1 and also lacks the 6 layers. That is why this is one of the reasons why this is called as the agranular cortex. Okay. So, and Campbell also further went on to classify this area, that is rostral to the central sulcus, into 2 sub areas. The more caudal of them he called as M1 or primary motor cortex. Within the motor area, the more caudal one is called M1, and the more rostral one he called as the premotor cortex. Okay. So, let us remember this is the first half of the 20th century, right. Immediately following this, or around approximately the same time and immediately following this work, Clinton Woolsey performed his studies, classical studies on animals, and used it to classify these areas, and he found that stimulation of certain areas that is just medial within the premotor cortex. Within the premotor cortex, the areas that are lying on the medial surface of the cortex also behave somewhat similar to the primary motor cortex. Hence, he called this area as the supplementary motor cortex. Okay. So, this is the medial region within the premotor cortex. So, within the premotor cortex, the more medial surface is called as a supplementary motor cortex. Okay. And what he found was that the supplementary motor area had less detailed motor maps. So, we discussed the case of motor maps in the previous class, and in comparison with M1, supplementary motor area has less detailed motor maps and signals both start and stop of movements, unlike M1, and can activate parts of the body that are on both sides. So, once again different from M1. Important to note is that this is again the first half of the 20th century, right. So, then onwards this kind of studies have continued, and classification has also continued, and it has resulted in this situation. The situation is the following: the area that is, so this is the central sulcus, So, that is the central sulcus, right. So, the area that is rostral to the central sulcus, that is shown in green here, that is the primary motor cortex. Okay. The area that is immediately rostral to the primary motor cortex is now classified as the supplementary motor area, right. Actually, there are this has further been, the original supplementary motor area that was first proposed by Clinton Woolsey has been divided into two areas. One of this is the supplementary motor area proper, or area F3, and the more rostral region is called as a pre-supplementary motor area. Now, the premotor cortex, that was earlier called as premotor cortex, was first divided into supplementary motor area and the other premotor cortex. Now, the supplementary motor area has a supplementary motor area proper and pre-supplementary motor area. Also, functionally distinct other regions have also been found. The dorsal convexity of area 6, so this is area 6, right. So, this is area 6. So, the dorsal convexity of area 6 has been divided into two parts. The more caudal region is called as the dorsal premotor cortex or area F2. Okay. The more rostral region is called as the pre-dorsal premotor cortex, that is area F7. The ventral region, the ventral convexity is called as the ventral premotor cortex. This has been further divided into two areas, F4 and F5. Okay. Now, the region that is caudal to the black line that I am now going to draw, I am going to draw a black line here. The region that is to the right of this, right, this is M1, this is M1. Okay. To the left of this you have the premotor area. In the ventral convexity you have the ventral premotor cortex, and the dorsal convexity is divided into two areas. The more caudal one is called as the dorsal premotor cortex, and the more rostral area is called as the pre-dorsal premotor cortex. Okay. Now, how has this division been made? Based on functions. So, these are functionally distinct regions. These regions, it turns out, have interconnections among them. So, there is interconnection between these areas, there is interconnection between these areas, there is interconnection between these areas and M1, there is interconnection between these areas and SMA, for example, and they also have interconnections with other regions, for example, the parietal cortex. Some of these regions do not have so much interconnections with the parietal cortex, but rather have interconnections with the prefrontal cortex. So, this presents a very confusing picture of the situation. So, we will take it slowly from here and try to understand what is the meaning of this. So, these premotor areas that are lying to the left of the red line here have interconnections among them, have interconnections with the primary motor cortex, and have connections with the other cortical regions. Now, that means, so that what are the kind of connections that these have? They have reciprocal connections. That means reciprocal means these regions connect to some other region, and there are neurons from the other region that connect to this region also. So, these are reciprocal connections, and some of these connections can be convergent. So, that means many neurons from a particular region could converge into a particular region, particular area in one of these premotor areas. So, several neurons from several distinct zones could converge into a relatively small area, right. So, several cables could come, for example, like this, right. Several neurons could come and converge let me redraw this. So, could converge into a small area here, but these could arise from different regions. So, that is convergent. They could also be divergent. So, projections from here that start from a relatively small area could go to different regions, to relatively larger areas or to functionally distinct areas. So, these projections are called as divergent connections. So, all these things exist. There are convergent connections, there are divergent connections, there are reciprocal connections, and also classification is also based on the crucial properties of neurons within this area. So, in other words, functionally these are different, but also properties of neurons within these areas are also different. So, that is one of one aspect of the connections within the premotor region. Now, also we have to note that the supplementary motor area proper, which is F3, the ventral premotor cortex is this is F4 and F5, okay, and the dorsal premotor cortex, that is F2, primary motor cortex. They have well organized projections with the primary motor cortex. Somatotopically organized means regions that correspond to a particular area of the body, particular part of the body, project to a particular part of the SMA or PMV or PMD. So, this somatotopic organization with projections with primary motor cortex is maintained with the ventral premotor cortex, with the dorsal premotor cortex, with the supplementary motor area. And also M1 and the supplementary motor area are different from the other areas in the sense that they have motor neurons that could activate the lower motor neurons. They have neurons that could activate motor neurons in the spinal cord. So, this is also called as the corticomotor neurons or the upper motor neurons. More discussion on that will follow in future classes. So, these neurons project directly to spinal cord, or some of these neurons project to spinal cord and cause movements. These have, or these areas receive somatotopically organized inputs from the primary somatosensory cortex. This is the area that is caudal to the central sulcus. The area that is rostral to the central sulcus is the primary motor cortex. The area that is caudal to the central sulcus is the primary somatosensory cortex. So, they receive inputs from the somatosensory cortex. So, what could that mean? That means information or sensory information about the state of the body is shared with the control, motor control area, or what is the current state of the body. How is that? How do we know that? From receptors, from sensory signals, from various afferent systems. This could be proprioceptors, this could be many other systems. So, this information is shared with the primary motor cortex and the supplementary motor area. Note that, but not with the premotor areas, whereas the premotor areas, which is the ventral premotor area and the dorsal premotor area, these also project to the parietal cortex, these also receive inputs from the parietal cortex, but from the more caudal parietal cortex. So, not from the primary somatosensory cortex, right. Critically, the following must be noted. So, what we have discussed so far is that supplementary motor area proper connects to the primary somatosensory cortex, this we have discussed. So, whatever we have discussed so far, I will mark in black or I will check with black. We have discussed the projections received by the supplementary motor area with the, so that connections, that the inputs it is receiving from the primary somatosensory area. M1 is also receiving from the primary somatosensory area. PMV and PMD, which are these and this is inputs from the more caudal parietal regions, right. Now, what is not discussed, I am going to circle with this blue, right, that is these areas, is it not? Pre-dorsal premotor cortex and the pre-supplementary motor area, these two areas on the other hand, in comparison with the other premotor areas, receive only weak inputs from the somatosensory cortex, but very strong projections with the prefrontal cortex. What could this mean? This means that the regions that are more caudal within the premotor area, which is these regions, right, receive inputs from the somatosensory system about the state of the body, whereas intentions. So, the prefrontal cortex is believed to be the seat of executive decision making. So, intentions, context specific control is probably achieved through the connections received from the prefrontal cortex through the pre-supplementary motor area and the pre-dorsal premotor cortex. Okay. These two do not directly project to M1 and they have reciprocal connections. So, they project both ways, bidirectional connections with the prefrontal cortex. So, this means the areas that are rostral within the premotor areas, which are these two, pre-dorsal premotor area and pre-supplementary motor area, these two define the context specificity of motor acts, whereas adjustments with respect to the sensory information is made by the dorsal premotor cortex, by the ventral premotor cortex and by the supplementary motor area. And direct information from the primary somatosensory cortex is received by M1 and the supplementary motor area. So, what experimental approaches have given us the information that we now know about the organization of the primary motor cortex and the premotor cortex? How do we know? What kind of experimental approaches have been developed? In the 1950s, Herbert Jasper and his colleagues developed an experimental approach to record from the primary motor cortex of live behaving animals. So, this is using microelectrodes recording. So, there are two things, one is stimulation using microelectrodes, the other is recording. So, the electrodes that are used for recording can also be used to stimulate, supply relatively small stimulating voltages for a brief amount of time. And what would be the effect of such stimulations on the motor activity? That is called intracortical. Intra means within, cortical means cortex, right. So, intracortical, within the cortex microstimulation, stimulation that is achieved using a microelectrode. So, this is intracortical microstimulation, that is one approach. The other approach is recording. When the animal is behaving naturally, performing some activity, which area is responding how, that is recording. So, these are all possible in animals and have been successfully, very successfully implemented in animals. How do we do this in human experiments? How do I record activity of behaving humans? What is going on in their brain? What is going on in their cortex? What is going on in their the motor area? And can I also stimulate selectively specific regions and see what the effect is? The answer is, in today's world it is possible to do these things with some limitations. What are these? How do you record? This approach to recording brain activity is called as functional neuroimaging or functional magnetic resonance imaging. This is called as fMRI, right. And how do you stimulate? If you want to stimulate, how do you stimulate? This is possible today using transcranial magnetic stimulation or what is called as TMS. Other techniques are also available. These are TDCS, transcranial direct current stimulation, transcranial alternating current stimulation, TACS. These techniques are not the exact analogues of the intracortical microstimulation and recordings that are available in animals. Obviously, in animals we could perform the surgery, implant the electrode and properly do the recording or stimulation. In humans, ethical considerations exist. So, we will have to do these things non-invasively. So, limitations come because of this reason. In fMRI, for example, we do not have the same kind of resolution that we would have with the recordings that we will get from microelectrodes, right. So, so that is one problem with fMRI. Again with the stimulation, once again you are not going to have the same kind of stimulation that you are going to have with the intracortical microstimulation with microelectrodes achieved in animals. So, this is different, but this is possible to some extent. We are able to get some analogues of what was earlier done in animals. Okay. So, this method was first championed by Herbert Jasper and his colleagues, right. So, and this work was later taken over by the famous Ed Evarts, where so he worked in National Institute of NIMH Bethesda, right. So, his contribution was in studying the activity of individual neurons for during performance of specific movements by live behaving primates. So, he took monkeys and in monkeys he recorded activity while this monkey was performing specific actions, specific movements. Suppose this monkey was performing flexion of the wrist. So, the wrist is here and performing flexion. So, the flexion of the wrist, right. So, what Evarts found was that, or extension of the wrist as the case may be, what Evarts found was that the neurons that are active during flexion are reciprocally silent during extension, and the neurons that are active during extension are reciprocally silent during flexion. So, in other words, the question is here, what is the relationship between movement parameters? So, what is the movement parameter? Here I am moving a particular part of the body, or I am performing I am changing the joint position, right. So, this movement is actually kinematics. Kinematics means, kinematics could include displacements, velocities, accelerations, etc., right. This could be linear or angular. Okay. The question is how is this kinematics achieved? Which particular set of neurons or which set of which sub area are responsible for this are the questions. How is this kinematics even achieved, or is there a correlation between the kinematics of the movement and the activity? That is the question. The answer is yes, there is some correlation. So, what he found was that, so let us suppose I am first drawing the kinematics of the movement. Let us suppose that flexion refers to going down in this plot like that, and then the animal is extending like that, right. This is the kinematics, this is flexion and if we go up we are extending. Okay. And now he recorded activity of neuron, of a particular neuron. What this neuron did was, it was silent. So, I am going to draw the neuronal activity in a different color, say this green, right. So, this is neuronal activity. This neuron is silent until about that time, say for example, and then starts firing during or just prior to extension and during extension. So, this time difference between the start of the firing of neuron to the start of the change in kinematics, how much would that be? That would be anywhere between 50 to 150 milliseconds. We have seen this earlier, right. We have discussed that these delays are the classical neurophysiological approach to studying movements, right. So, this means anywhere between 50 and 150 milliseconds before the movement onset, this neuron starts firing, right. So, that means this and it stops again some time before, actually there is a gap after the stoppage of activity of this neuron, but the movement continues for some time after. Okay. So, this is about 50 to 150 milliseconds. Actually this area, this particular topic continues to be still actively researched. There is still debate about what is being performed in M1, what exactly is the role of M1, this is still being actively researched, right. So, there is a raging controversy on what does the primary motor cortex encode, right. We will continue to discuss based on the previous studies, although this is being actively researched, we should continue to actively learn about what has been previously done. Okay. So, it turns out that at least what the following is generally known. What is generally known is that the premotor area in general encodes context, as one would expect, and the primary motor area or M1 encodes the mechanics. Exceptions to this particular statement exist. In fact, there are just too many exceptions. However, it is now widely believed that the premotor areas are responsible for setting the context of movement, whereas the primary motor area is what encodes the mechanics. So, when we say mechanics, there are several things. One is kinematics, which defines movements, but how are these movements themselves caused? These movements are caused by forces that are produced, right. So, these movements are caused by the action of muscles, by contraction of muscles. So, that is actually kinetics. So, there is both kinematics and kinetics. Now, a question is what does the primary motor cortex encode? If the primary motor cortex encodes the mechanics, does it encode the kinematics or does it encode the kinetics? That is the big question. As I said, considerable amount of debate exists about what is actually being encoded by the primary motor cortex, but here we are discussing the work of Eddie Watts. Okay. So, what Eddie Watts did in a different study, so I suppose this study was from the 1950s, the one that I just explained was from the 1950s. Later, I think in the 1960s he did a different study, which is so this is the 1960s study, I suppose, please check that. So, where he said the kinematics of a particular movement, he designed the experiment in such a way that the kinematics of a particular movement remained constant, but the kinetics would change depending on the direction of a load. If he kept through a system of pulleys and ropes, he kept loads that could either assist flexion or, so when I am doing that movement I am flexing. So, if a load, that load is hung in such a way that assists my flexion and opposes extension, right. So, when the load is hanging in such a way that it assists flexion and opposes extension, or the load could be hung in such a way that it assists extension and opposes flexion. So, and he actually recorded the wrist angle using what was available at that time, using a generic potentiometer. So, and he, of course, the rest of the body parts of this animal were restrained from moving. Obviously, you do not want to perform the same movement with other muscles, and he recorded from the wrist area of the primary motor cortex. Okay. And so, and note, the task was always the same. The task is to perform a particular joint motion or a particular change in joint angle, or the kinematics remained the same. Crucial to this experimental design is that the kinematics remained the same, whereas in the case where the load assisted flexion, flexion was easier to perform, is it not? In the case where the load opposed flexion, flexion was harder to perform. An expectation would be that, you know, if the system does not care for what the outcome is, it only cares for the kinematics. If it does not care about the actual environmental situation, whether it is opposing the flexion or supporting the flexion, then you should have practically the same activity in both the cases, right, because the joint angle that is being moved is still the same, the kinematics is still the same. So, you should have the same activity is the expectation. What was found, that is the question. What was found was the following. So, first and so here is the case where the weight opposes the flexus, and here is the case where the weight assists the flexus. So, here are the diagrams that show the two, right, in these two cases. Now, let us see what actually happened. Okay. I am going to draw first the kinematics. So, here I am drawing the kinematics in red color, and note this kinematics is the same for all the cases. Okay. So, let us say that the movement happened at around that time, and so all the recordings are with respect to this movement onset. Okay. So, let us say that is extension of the wrist, and then if this kinematics goes up, if this curve goes up, that is considered flexion. Okay. Now, note that, you know, this kinematics is the same in all the experimental conditions, right. Now, what are the various experimental conditions? The first experimental condition is when there was no load, that was either supporting the flexion or opposing the flexion, there was no load. This is also called as a no load condition. In this case, what happens? He recorded simultaneously the EMG activity and the activity from the M1 neurons that control the wrist. Okay. So, what was found in the EMG case, when there was no load, so this is EMG of the flexors, right, there is flexor activity and then there is minor. So, there is some baseline activity here and then there is great activity during flexion. So, this is flexion, is it not? During that time there is flexion, and after some time there is reversal back to the original baseline activity. This is the flexor activity. Okay. In the case of extensors, there was activity just before the onset of movement, but as soon as the movement was onset or just before that, there was the extensor activity went to baseline. This is the extensor activity. This is in the no load case. Now let us take the case when the load assisted fluxion, and we said what would happen if the expectation is that the system cares only about kinematics or movements, but not about kinetics or forces, then what you would see is practically the same activity similar to these would be observed, whether there is no load or resistance or opposition. In all cases what was found, and by the way what was the neuronal activity, that also we need to draw. So the neuronal activity is drawn in green here. The neuronal activity is the following. So here each vertical line denotes an action potential from these neurons. Okay. So there was activity just some time before, right, and then after some time the activity reduced to a baseline. So this, what happens when during the load, when the load assists flexion, what is the EMG activity? In this case note the load assists flexors, is it not? In this case the flexor activity is nearly baseline sometime before the movement onset and continues to be nearly baseline. In other words, the monkey is taking advantage of the fact that the load is assisting the flexors. Anyway I want to perform flexion movement, and because this is flexion movement, is it not? Anyway the kinematics is going towards flexion, is it not? We said this is flexion, this is extension. In that case, since the load is assisting my activity, flexor activity is baseline throughout, right. And what about the extensor activity? This is the flexor activity or EMG, this is EMG. The one in green refers to neuronal activity, right. What about the extensor activity? The extensor activity is present just before the movement onset, until just before the movement onset, after which it becomes baseline during the movement, is here, right, and then again it starts. Okay. This is the extensor activity. Now there is one more case, this is the last case, that is the case when the load opposed flexion. Now what happens? Now what about the EMG? Also we have to discuss what happened with the neuronal activity during when the load assists flexion. Practically, since it is the flexion is assisted by the load, practically the activity of the neuron is baseline, there is not any particular activity. So when the load opposed flexion, what happens to the EMG of the flexors and the extensors? What happens to the flexors is, before the onset of movement there is activity, and note at the onset of movement you realize that the load is opposing you, so you need to activate more to perform the flexion. So what happens is that the activity goes up, the amplitude actually goes up and then reduces to a smaller level, very different from the baseline case, right. And what this is for the flexors, and for the extensors it was nearly baseline. This is for the extensors. Now what about the neuronal activity? The neuronal activity is that just before the onset of movement there is great activity here, and this activity continues, during movement and slightly reduces later, but does not go to baseline. So, from this what we have learnt? From this we have learnt that, or at least this data, or the data from Ed Edwards has supported the hypothesis that for the same kinematics, for the same movement, depending on the kinetics, depending on whether the load assists or opposes a particular movement, activity in the, not only the muscle, obviously the muscle activity will change because that is going to cause a mechanical action, not only the muscle but also the neurons will change. So, this was a very strong evidence in support of the hypothesis, in support of the idea that the cortical neurons or the M1 neurons encode the kinetics of movement. Okay. What we have seen so far is the contributions of Herbert Jasper and Ed Edwards. So, thank you very much.